The process by which we manufacture the formed elements of the blood is called hematopoiesis. And, there are three subtypes of hematopoiesis. There's a process for making the red blood cells component, there's a process for making the white blood cells component, there's a process for making the platelets. And if we look embryonically at where these events occur, in embryos, and fetuses, the manufacture of these formed elements of blood first starts out in the yolk sac, in a location in the yolk sac called the blood islands, and that's about around the 3rd week after conception. And these blood islands eventually produce the stem cells that go out to other hematopoietic tissues in the body, like the bone marrow, the thymus, the liver, and the spleen. Now, the liver and the spleen also help to manufacture the formed elements of the blood, but only during the fetal development time. Okay, so the liver, for example, helps make red blood cells, but it only does so until about the time that the baby's born, and then it stops. The spleen also helps to make red blood cells up until the time of birth, and then it stops. It does, however, continue to assist in the formation of white blood cells throughout the person's lifetime. After birth, the main place where hematopoiesis occurs is principally in the bone marrow, with some assistance, as I said, from the spleen. And we'll also see the thymus is important in part of white blood cell development in adults. So let's take a look at these 3 different types of hematopoiesis. As I said before, there are 3 types of hematopoiesis. There's the one relating to the manufacture of red blood cells, the one for the manufacture of white, and the one for the manufacture of platelets. The manufacture of the red blood cells is called erythropoiesis. And erythropoiesis is a process that's has a very high productivity level. In fact, you can make somewhere around on the order of 2.5 million new red blood cells every single second in your bone marrow. Red blood cells, or erythrocytes, can live for roughly 4 months, or about 120 days, at which point they usually get screened out or removed by the spleen. The time it takes to actually make a mature erythrocyte is somewhere on the order of 3 to 5 days, depending on the health of the individual and whatever demands are occurring. The process by which we make a red blood cell starts with a stem cell, something called a hemocytoblast. And that stem cell undergoes mitosis, and it produces 2 types of cells: another stem cell, and then a differentiated cell called a proerythroblast. And at this point, we have to have the encouragement of a special type of hormone called erythropoietin, or EPO for short. And if erythropoietin is present, and if erythropoietin, by the way, is made by the kidneys, if erythropoietin is present, it stimulates then that proerythroblast to further differentiate in the bone marrow and become something called an erythroblast. And you'll remember from 201, blast means immature cell. So an erythroblast is literally an immature red blood cell. And then we go through a number of steps, and actually this picture here doesn't even show all the steps, but there are several steps involved in the process as the erythroblast matures further into what's called a normoblast, and then something really bizarre happens. The normoblast ejects its nucleus. It gets rid of its nucleus, and now we have what's called a reticulocyte. It's called a reticulocyte because it has a high amount of rough endoplasmic and smooth endoplasmic reticulum present. That reticulocyte is busy manufacturing millions and millions of copies of hemoglobin. By the time it's done, it's manufactured about 280 million copies of hemoglobin. And finally, once it's all done with that, we have a mature erythrocyte. At that point, it's released from the bone marrow and it enters into the blood. So, mature erythrocytes enter into the blood. Up to that point, everything that I talked about is actually occurring in the bone marrow. process of white blood cell formation is called leukapheresis. And leukapheresis also begins in the bone marrow, just like erythropoiesis does. And in this case, once again, we have a stem cell, and that stem cell, or hemocytoblast, gives rise via mitosis to two different lines of differentiated or committed cells, something that are called B progenitor cells and something called T progenitor cells. Actually, it gives rise to three. And something called granulocyte macrophage colony forming units. Say that 3 times fast. So these 3 different derived cells from the stem cell will eventually give rise to 2 different types of lymphocytes called B lymphocytes and T lymphocytes. And then these guys here, the granulocyte macrophage colony forming units, give rise to things like the monocytes, the neutrophils, the basophils, the eosinophils. Again, this is all initiating in the bone marrow. And if we were to look closely at the surface of these different cells, we would see that they have different types of hormone receptors on their surface. And those receptors are for colony stimulating factors, which is a whole class of hormones that helps determine whether a progenitor cell becomes a eosinophil or whether it becomes a B lymphocyte. So depending on what type of colony stimulating factor receptor I have, I'm going to respond differently to different hormones secreted. Usually these colony stimulating factors, these hormones, get secreted when a person is faced with an infection. For example, if I'm faced with a bacterial infection, the type of colony stimulating factor that's going to be released is going to stimulate some cells to become neutrophils, because neutrophils are very aggressive bacterial consumers. If, on the other hand, what I'm experiencing is an allergic reaction, then I'm going to be stimulating those cells that eventually develop into eosinophils, because eosinophils are implicated in allergic reactions and also in things like infections with certain types of parasites like roundworms. So again, depending on the type of colony stimulating factor released, I'm going to get only those cells with a receptor to that type of colony stimulating factor to respond and begin developing into a mature type of white blood cell, whatever that white blood cell might be, the monocyte, a B lymphocyte, a T lymphocyte, etc. By the way, these B and T progenitors end up being part of what's called the lymphoid cell line. They are the ones that give rise to the lymphocytes, the B lymphocytes and the T lymphocytes. We're going to learn a whole bunch about the B and T lymphocytes when we get to the immune system. The rest of these, the granulocyte macrophage colony forming units, give rise to the monocytes, neutrophils, eosinophils, and basophils. The third subclass of hematopoiesis is the thrombopoiesis, and this is the process by which we manufacture platelets. Now, I want you to know that in old textbooks, they used to call platelets thrombocytes. That's the old word. Remember, cyte means cell, thrombo means relating to blood clotting. When it was realized that these platelets were not true cells in themselves, but actually little pockets of cytoplasm sloughed off from a larger cell, they realized, well, we can't call them thrombocytes because they're not a true cell. They do not contain a nucleus. They do not contain all the organelles associated with a cell. That's when they changed the name to platelet instead. But the process of formation of these is still called thrombopoiesis because thrombo relates to clotting. Poiesis is the formation of these clotting factors. And the cell that gives rise to all of these little cell particles that get sloughed off that are called platelets are these things called megakaryocytes or megakaryoblast. And these are giant cells. They're huge. Look at, here's a red blood cell and here's a white blood cell and here are platelets, and look at what their size compared to this megakaryocyte here. Megakaryocytes get that big because what happens is a megakaryoblast, the immature cell, undergoes mitosis but it does not undergo cytokinesis. Remember, mitosis is how we divide up the nuclear content of a cell. Cytokinesis is how we then divide up all the rest of the cytoplasmic contents. So if I keep going through rounds of mitosis, but I don't undergo cytokinesis, what happens is I start accumulating nucleus after nucleus after nucleus after nucleus, and these guys can actually have as many as 32 nuclei present inside of this giant cell. And then that giant cell starts to secrete off little packages of cytoplasm that are filled with clotting factors. Those little packages of cytoplasm filled with clotting factors are the actual platelets. So they're like little flakes off of the big cell. I like to think of them like a little shopping bag filled with clotting factors that then get released and circulate around in the blood. They only live for roughly about 4 days, so we have to have lots of megakaryocytes constantly manufacturing new platelets and sloughing them off. And these guys are found in the bone marrow. So the process of thrombopoiesis occurs in the bone marrow, the initial steps of leukopoiesis occurs in the bone marrow, and erythropoiesis also occurs in the bone marrow.